including conventions, norms, and institutions, evolve, perhaps improve, over historical time. Call this the transmittive dimension of culture. The outcome is that virtually all of humans' most remarkable achievements, from steam engines to higher mathematics, are based on the unique ways in which individuals are able to coordinate with one another cooperatively, both in the moment and over cultural historical time. But this explanation of human uniqueness in terms of cultural processes creates another puzzle, and this one is not yet solved. In this case, the focus is not on the level of the species and its achievements, but rather on the level of the individual and its psychology. How do human individuals come to the species' unique cognitive and social abilities necessary for participating in cultural coordination and transmission? To answer this question, the obvious first step is to establish exactly how human psychology differs from that of other primates, precisely how humans as individuals are unique. The difficulty is that over the past few decades, empirical research has established that humans' nearest living relatives, the great apes, possess cognitive and social skills highly similar to those of humans, including many that are seemingly relevant to cultural processes. For example, there is recent research demonstrating that at least some great apes, 1. Make and use tools, 2. Communicate intentionally, or even linguistically, 3. Have a kind of theory of mind, 4. Acquire some behaviors via social learning, leading to culture, 5. Hunt together in groups, 6. Have friends with whom they preferentially groom and form alliances, 7. Actively help others, and 8. Evaluate and reciprocate one another's social actions. But do apes do these things in the same way as humans? To make this determination in particular cases, we must look beneath the sweeping claims that both apes and humans have X or do Y, even though such claims may be true on a general level. To penetrate beneath such generalities, we need to make more fine-grained comparisons by performing comparative experiments in which humans and great apes, especially chimpanzees and bonobos, as humans' nearest living relatives, are observed in as similar as possible circumstances. Such controlled experimental comparisons make it possible to detect subtle differences of behavior and, ideally, the cognitive and motivational processes underlying them. In this way, we seek to identify the differences on the individual psychological level that ultimately lead to humans' unique forms of cultural coordination and transmission, and so to telescopes and parliaments. Given a description of the key differences between humans and their nearest great ape relatives, the next task is to explain those differences. In an evolutionary framework, the axiomatic explanation is, of course, natural selection. The human individuals alive today have been naturally selected to meet certain species-unique ecological or socio-ecological challenges. For example, one proposal is that humans evolved many of their unique cognitive and social capacities in response to ecological challenges that first forced them to collaborate with one another in acquiring food and then later prompted them to form larger cultural groups to defend their resources from other groups. Under these conditions, individuals who could best cooperate with others, individuals who were both capable and motivated to put their heads together with others to collaborate or form a culture, were at an adaptive advantage and so proliferated. But natural selection creates nothing. Natural selection is only a sieve that sorts, after the fact, viable and non-viable organisms. Evolutionary novelties originate not from natural selection, but rather from the other main dimension of the evolutionary process, inherited variation. Classically, inherited variation in evolution emanates from genetic mutation or recombination, which produce via ontogenetic processes novel traits. But recent advances in evolutionary developmental biology, so-called evo-devo, suggest that the constructive role of these ontogenetic processes has not been fully recognized. Not only do new traits always come into existence via ontogenetic processes, which direct and constrain genetic expression, but by far the most frequent source of new traits is changes in the timing and manner in which already existing genes are expressed and transact with the environment. Thus, even relatively modest changes in the way that regulatory genes orchestrate ontogenetic timing and plasticity can have enormous and cascading phenotypic effects, not encoded directly in the genes, as developing systems interact with one another and with the environment in unexpected ways. The implication is that if we wish to explain how uniquely human psychology is created, we must focus our attention on ontogeny, and especially